Thank you for joining us for the next in a series of videos that discuss the challenges of treating high-grade glioma. Be sure to visit the website shown on screen to see all the videos and access additional content. You see, it used to be that we studied high-grade gliomas by looking at a cell that's cultured from the patient or an animal model. With the advent of genetic tools, we could actually study glioblastoma and high-grade gliomas in its native form to see what it actually is in a patient. And what we learn from that is almost every cell in the tumor bulk is actually different. And what that means is that it's unlikely that any single agent will be able to destroy the bulk of the cell, since essentially every cell within a tumor is different.
Yeah, when you really think about this, there's all these different separate little microenvironments and all these packets of cells that are acting in different ways. So there's hypoxic areas of the tumor. There are tumors with high blood flow and lots of oxygen. There are areas that you have an intact blood-brain barrier that's going to exclude some drugs from getting in, and other places where it's broken down. And you have areas that are proliferating, actually growing, and other cells that really aren't dividing so much as they are sort of migrating out into the surrounding brain. And so you have all these competing factors and all these different combinations that make this diverse cellular population that's really hard to treat.
So certainly for radiation plays an important role. Certainly having a hypoxic core to that tumor leads to radiation resistance, and it requires other drugs to help improve the effectiveness of radiation. An important part of the tumor heterogeneity is really that idea of a moving target, that these tumors also change over time. And so even though you do a biopsy and find a mutation, once you treat that tumor, actually it will change and develop new mutations. And without having additional biopsies, we're really treating the wrong target.
The complexity of the cell-to-cell variation and the different microenvironment really speaks to the need to engage the immune system. Our immune system is designed so that it could tackle different things in different contexts. And if we could engage our immune system in a way that could attack all these different contexts and cells in high-grade glioma, then we have a chance of beating it. Tumor heterogeneity must be overcome if we hope to be successful in managing high-grade glioma.